All right, so here on twists and hydrogen bonds break, and so that's what we see here. The first thing is remember that DNA is what is called anti-parallel, meaning that their ends are opposite, that we call them 5' and 3' ends. So this side on the top here is a 5' end, this is the original 3', and then opposite here 3', so this is the 5' here. All right, and so what happens is that this little guy here, all right, where they come apart, so I call them unzipped, the hydrogen bonds are broken between your two sides here. You have this Y-shaped section of your DNA. Does anybody remember what that's called? It's called a replication something. Fork, all right, so a replication fork, kind of like a fork in the road, all right, there's like two different directions here. All right, that's called the replication fork, and then you have these enzymes. So enzymes, they're called DNA polymerase 3. They add nucleotides, but the thing about the DNA polymerase 3 is that they can only add in new nucleotides in a certain direction, and that is, if we look here, this little guy, notice here the arrow's going this way. It added a nucleotide, and so on and so forth, and it continues adding the nucleotides. So the new strand grows in the 5' to 3' direction. So that's what we have here. So it started out on the 3', started adding in the 5' direction. This side is called the lagging strand, and the reason why is because it cannot be built in a continuous strand. So you have to imagine that, let's say, as the DNA unzips, it unzips a little bit at a time. Let's say at the beginning it only unzips and separates this far. So what happens is the DNA polymerase 3 binds here, adds nucleotides in the 5' to 3' direction. It can't bind to the end here because the new strand, this is going to be the 3' end, to be opposite here. So it has to start at the 5' end and move towards the 3' end. So therefore, DNA polymerase adds nucleotides in this direction till it gets to the end. Then what happens? It unzips some more. No problem for this side. The DNA polymerase just keeps on adding nucleotides in the 5' to 3' direction. But here it got to the end, and now it has to start over again and grow in the 5' to 3' direction. So what you end up with is having fragments. So you get these fragments here, and so now it unzips some more. It's showing you unzip some more. The top strand here keeps on going, and you get one big piece here. At the bottom strand, you've got all these little fragments, and so it'll build it to this end, and then it'll come over here and build another fragment, come over here and build another fragment. Those fragments are called Okazaki fragments, based upon the Japanese scientists who discovered them. And so that's labeled up here. All right, and so this is called leading strand. The leading strand is always the one that's continuous. The lagging strand has the fragments. So the problem is now you have fragments instead of one long continuous strand, so you have an enzyme called DNA ligase to glue them together. All right, so what I'm going to have you do in the video, right before this picture, there were 3 bullets that I didn't do. I'm going to have you go back to the 3 bullets previous to this picture, and from what we've talked about, you should be able to fill in those blanks.
I just need to clean this real quick. Thank you.
So you should have, for the first one, along one template strand of DNA called the leading strand, DNA polymerase can synthesize a complementary strand continuously, all in one big piece. And so if we go over here, that's what we see here. All right, it just keeps on adding, and even as it unzips, it just continues on adding, and you get one big strand. So that's the leading strand. Whoops, wrong way. And it's going towards the replication fork, remember, which is that where that Y shape there, so it's moving towards it. All right, and then the other strand, to elongate the other new strand called the lagging strand, DNA polymerase has to work in the direction away from the replication fork in order for it to be in the 5 to 3' direction. So that's what we see here. So it's moving, it has to come over here and restart all the time at the replication fork to go from the 5 to 3' direction of the new strand. Whoops, going the wrong way. And so that lagging strand is synthesized in fragments, and we call those Okazaki fragments, which then will be glued together by the enzyme ligase. All right, and so how'd you guys do on those? Good? All right. Okay, so one thing, we're going to add another thing onto this, all right, is the fact that actually before all of this can happen, the DNA polymerase can add new nucleotides to build and replicate your DNA. The DNA has to be what's called primed. So this section is called priming DNA synthesis. So DNA polymerases, notice that it says polymerases. There's a whole class of these enzymes called DNA polymerases. So there's DNA polymerase 3. We're going to learn about a DNA polymerase 1. So there's a whole bunch of them. They cannot initiate synthesis of a polynucleotide. Poly means many, so it's hooking together many nucleotides together to make your DNA. They can only add nucleotides to the 3' end. So what does that mean? If I were to draw this here like this, here's my DNA strand. All right, so I've unzipped it here. All right, and let's say this is the 5' and this is the 3' end here. Oh, let me keep it consistent with the last picture. This would be the 3' and this is the 5'. All right, so when I build my new strand, that complementary to this is going to have to be the 5' end, all right, to be anti-parallel, and this will have to be the 3' end, right, to be anti-parallel. Which one is going to be the leading strand? Top or bottom? The top, to grow in the 5 to 3' direction. So when I build this new strand here, this end would be the 3' because this is the 5'. All right, what it's saying is that back in this picture here, is that it looked like that DNA polymerase just started at the end and started adding nucleotides. And realistically, that's not what happens. DNA polymerase, if there's nothing there, it can only add to a pre-existing nucleotide, and at the end of the DNA, if it's at the very end of the DNA, there's no pre-existing nucleotide. So DNA polymerase can't add anything, and so replication can't happen here. So therefore, you have to have a primer, and oftentimes it's RNA. And there's a special enzyme called primase that adds an RNA primer here. So we add an RNA primer, and then you have nucleotides there, and then your DNA polymerase can now, since there's pre-existing nucleotides, can add nucleotides and grow in the 5 to 3' direction. But it can't do that unless there's a primer there first. All right, so then let's look at what's the problem with the lagging strand down here. So remember, it unzips a little bit at a time. So let's say it unzips here all the way to here first. All right, so this adds one primer and starts adding nucleotides. Here, where's the primer going to be added? It's not going to be added at the end because it has to grow in the 5 to 3' direction. So the primer would be added here, all right? So here's your primer. And then now DNA polymerase can grow in the 5 to 3' direction to the end. Then the DNA unzips some more, and we have to build another Okazaki fragment, so you get a new primer. And then now DNA polymerase can add the nucleotides and so on and so forth. And so as it keeps on unzipping, each fragment has to have its own primer. So that DNA polymerase, which does what's in blue here, can add nucleotides. All right, and so that's what the next few pictures are going to show in this next couple of bullets here. So the initial nucleotide strand is a short one called an RNA or DNA primer. It's usually RNA. And so that's what I put here in red. That's my red up here on the board. And what does that is an enzyme called primase. So primase starts the RNA chain from scratch. So it doesn't need to have pre-existing nucleotides. And so therefore that's why it works. All right? Whereas DNA polymerase does. So only one primer is needed to synthesize the leading strand. Here, draw on the board here. But the lagging strand, each Okazaki fragment has to be primed individually or separately all by itself. So realistically, to kind of go with this, if we go back to this picture here, to make this more complete, you could add, like I did here, primers, all right? So I can't draw here, but I would add, if you're doing color coding here, I would add one primer here and just color the end here. This one here, each one of these fragments has a primer, so like this. So this would make it more realistic, and that would be red here. So therefore, in this picture here, you have a primer here, right, at the beginning, and a primer at each of the beginning of each of these Okazaki fragments. So you have these primers here. All right, primer. So I'm going to write that here that these are primers, RNA primers. Added by what? By primase. So that makes this picture more realistic about what happens. Okay. So the next series of pictures, I like this picture better, but the next series of pictures shows you just one side of this. This shows you both the leading and the lagging strand. The next series of pictures just takes one, and it's the lagging strand, one side of this, and shows you what happens. So let's look at that. All right, so we're just looking at, in here, we're just looking at the lagging strand. So it'll be just like this part right here, okay? So, so you have here, this is the beginning of your Okazaki fragment, so this is your primase. So that top picture is like the beginning here. So you're adding your primase, primer and the enzyme primase does that. And then after that primer is added, then DNA polymerase 3 can start adding the nucleotides. So that's like what I did here in blue. So it's adding the nucleotides till it gets to the end. And this is your first Okazaki fragment, just like this one right here. All right. Then it has to go back and start again and make a new Okazaki fragment. Again, something I don't like about this picture here is it doesn't have the primer for the new Okazaki fragment. So I'm going to add that in there. So that's your red primer there. So that's like this strand here that I just drew. You're adding a new primer. Then DNA polymerase can add nucleotides to the end of that first fragment. Okay. Then what we haven't talked about yet is this little yellow guy here. This is DNA polymerase 1. It's another type of enzyme in this class of DNA polymerases. Polymerase 3 added the new nucleotides. If you look here, it says DNA polymerase 1 replaces the RNA with DNA. Do you see that at the end, you're going to have DNA, this DNA strand after you finish, and this DNA after you finish is going to have some RNA pieces in it, and this is going to be part RNA, part DNA. We want our DNA to be all DNA, not RNA. So it replaces them out. Let's talk about what's the difference between them. So let's compare and contrast. We've talked about this before, but I think we need to review it a little bit. All right, so this replaces the RNA with DNA. So let's look at and compare RNA nucleotides with DNA nucleotides. what's the difference between them? There are 3 parts to a nucleotide. 3 parts to a nucleotide. Can you remember what they are?
It's like pentose sugar, phosphate group, or some phosphate, and then that's all I got.
Okay, so you have a sugar, the pentose sugar, and you have a phosphate group. Anybody remember there's one other part, Grace?
Nitrogenous bases.
Nitrogenous base, and that comes off here. All right, so I'm just going to put an N base. N stands for nitrogenous. That means it's a base that contains nitrogen. All right, those are your 3 parts of a nucleotide. And so both RNA and DNA nucleotides have those same 3 parts: the sugar, the phosphate group, and the nitrogenous base coming off the sugar. What's the difference? Do you guys remember what's the difference between the nucleotides in DNA versus RNA? And it's represented in the name. Grace?
DNA has deoxyribose sugar and RNA just have ribose sugar.
That's right. So the first difference is in the sugar. So DNA, the D in DNA stands for deoxyribose. So here the sugar is deoxyribose. And here the sugar is ribose. So they're very similar in structure, except deoxy means it's missing an oxygen. So it's ribose without an oxygen. All right? And so the sugar is different. They both have a sugar, but the type of sugar is different. What's the second difference in the nucleotides?
The nitrogenous bases are different, or one of them's different.
Okay, that's right. Nitrogenous bases are different. Let's do DNA. What are the nitrogenous bases in DNA? Anybody remember? Yeah, this is your A, T, C, and G. Adenine, thymine, cytosine, and guanine. All right? 3 of those are the same in RNA, and 1 is different. Can somebody tell me what RNA has that DNA does not have? A U, that's right. It has a U. So here we have A, and instead of writing a T, they have a U and a C and a G. So therefore, there are 4 different nucleotides for RNA. All RNA nucleotides have a phosphate, ribose, and then they could have an A, a U, a C, or a G attached to it. And there are 4 different kinds of DNA nucleotides. They all have a phosphate and a deoxyribose, but the base could be an A, a T, a C, or a G. Yes? Yeah. All right, so when it says, so I want to review this. So when it says DNA polymerase 1 replaces the RNA with DNA, all right, so therefore what's happening is, let's say we have, let's say here's your primer. All right, let's say there's an RNA nucleotide right here, and the base sticking out is a U. All right, so it has a phosphate, ribose, and a U here. All right, bonding with, what would be the DNA base here that would bond with? What would go with a U? An A. U replaces T. A U replaces T, so you have to think of what would go with a T. All right? So therefore, the DNA would have an A here. So you have an RNA nucleotide here. And so when it replaces it, what does this mean? DNA polymerase 1 is going to pluck out the RNA nucleotide and replace it with a DNA one. The DNA one would have a what? Which one DNA nucleotide would replace that to go with the A on the DNA? A T. All right. So plug in and make it AT, and then replace RNA with DNA. So big picture here, what RNA polymerase over here is doing, all right, is the primer is being taken away here. So it takes away the RNA, and then it's going to add the appropriate DNA. All right. Take away the RNA primer, add DNA. All right.
Why does it just start with like a DNA prime?
Why doesn't it?
Yeah.
Yeah, there are some cells that they do have a DNA primer. Our cells happen to be RNA primers, so it's just the way it is. I don't know why. All right, so so then you see that now you have all DNA, and now what can happen is ligase can come and glue it all together. So you have this fragment, ligase comes, connects it, and you've got one continuous fragment. Okay, and so that takes care of the lagging strand. Okay. So proteins that kind of overall that assist DNA replication. All right, so at the very beginning you had helicase, untwists and unwinds and breaks those hydrogen bonds. So that's called DNA helicase. Something we haven't talked about is once you untwist it and unwind it, you don't want it to go back together. So there's these what are called single strand binding proteins that bind to and stabilize the DNA so that once it's untwisted and broken apart, the hydrogen bonds, then you don't want it to go back together. You want it to stay apart so that you can add the nucleotide. So these guys I'll show you a picture in just a second, kind of keep the two strands from going back together. And then there is topoisomerase. This corrects overwinding ahead of the replication fork by breaking, swiveling, and rejoining DNA strands. Basically what this does, like here, ahead of the replication fork is right here. So helicase is going to untwist it and break it apart, all right, so that you can continue DNA replication. Do you see that if you untwist something, that you could untwist it too much and it'll start twisting the other way? That's the overwinding is talking about. So make sure that that doesn't happen. The next set, I think 4 of them, you should be able to answer based upon what we've talked about. So I'm going to have you try and answer those. Okay, so let's see how you guys did. So primase synthesizes an RNA primer, all right? And at the 5' ends of the leading strand, so one on the beginning of the leading strand, and at the beginning of every Okazaki fragment. DNA polymerase 3 continually synthesizes the leading strand, and then will also elongate each Okazaki fragment. Then what removes the primer? DNA polymerase 1 removes the primer of the 1' at the end of the 5' strand and at the beginning of every Okazaki fragment and replacing it with DNA. All right. And then lastly, DNA ligase joins the 3' end of the DNA that replaces the primer to the rest of the leading strand, and also joins all of the lagging strand fragments. All right, so what you end up having is two strands that are all DNA, all right, and continuous. All right, and so that's what this picture here shows you. So this picture here shows you all of that going on together. So here's your leading strand, and so this would be the 5' end, this is the 3' end of the new strand, so it's going and adding continuously in the 5' to 3' direction. Here is showing you bit by bit as is opened, you get the different Okazaki fragments, and you can see that the newest Okazaki fragment being formed is right here at the replication fork because it unzipped. All right, you have to imagine over time to make this Okazaki fragment, it was unzipped here, then it unzipped here, and so it had to be made in this direction, unzipped here, in this direction, and so on and so forth, so you get all these fragments. Notice that the leading strand, just one big strand, it just keeps on motoring on. here, the oldest fragments, you can see that they're in different stages of what we've just talked about. So the newest fragment is just being built. It's at the replication fork. So primase is building the RNA primer, and so it will be built until it gets to the last Okazaki fragment. While that's going on and building the new fragment, then you have over here, the primer was already built. Now DNA polymerase 3 is adding nucleotides to build the rest of this Okazaki fragment. While that's going on, this one has already been built, so you have the primer and you have the whole strand here. And then this one has also been built, but notice here the primer is really short. So DNA polymerase 3 is doing its job and taking out the RNA and putting in DNA nucleotides to replace it. And down over here, this is the oldest Okazaki fragment. The primer has already been replaced, and DNA ligase is gluing it together. So it's all kind of happening simultaneously, all right? And so therefore, eventually, all of those things will happen to each fragment, and eventually you will have one continuous strand here. And so what this picture up here, I just want to point here out this little box here shows you that what you're looking at is this section of this. But realistically, we don't start replication of our DNA at the ends and go from one end to the other end. So if you recall from the previous picture that I talked about in the video, let me just pull it up here to relate to it. This picture right... This picture right here shows you that this is realistically how DNA is replicated, is that, so we have one DNA molecule. Instead of starting here and just going from right to left, or starting here and going from left to right, we have these different what are called replication bubbles, where the DNA separates here, and then replication occurs in this direction and this direction. So there's two replication forks, and then they keep on unwinding and unzipping, and eventually they will meet, and you end up with two DNA strands at the end. So what this does is speed up replication for us. It would take a lot longer for DNA to replicate if you started at one end and just moved to the other end, all right? So therefore, this, by opening up the DNA in several spots and moving in both directions and allowing those to meet, makes it happen a lot quicker, all right? And so, well, let's look here. Let's go back to the... All right, so that is replication in a nutshell. The last two things I want to talk about is when something goes wrong. So proofreading and repairing DNA. So when things go wrong, DNA polymerase proofreads newly made DNA, replacing any incorrect nucleotides. What does that mean? So let's say your original DNA, let's say there's a nucleotide in this spot that had an A sticking out here. So my new nucleotide that DNA polymerase was supposed to add is supposed to have a T, right, to go with that A, all right? Sometimes they screw up and put the wrong one in there, all right? So sometimes they'll try and put a C with that A, all right? And that's not supposed to happen, so that's going to mess up your DNA. That's a natural kind of mutation, a natural change in your DNA. So most of the time, these DNA polymerases, these enzymes, correct that and get rid of the messed up one and put it in the right one, all right? So that's what it means by proofreading and replacing any incorrect nucleotides. This is called mismatch repair. Mismatch is when they accidentally match up the wrong base, all right? And so repair enzymes correct those errors in base pairing. Sometimes there's some other damage that can be done with DNA where it actually causes, we have to do what's called a nucleotide excision repair. Excision means to cut out. And what are we cutting out? We're cutting out the nucleotides. Why is it called repair? Because we're repairing damaged DNA. All right, so you're cutting out nucleotides that are, something's wrong with them. So enzymes cut out and replace damaged stretches of DNA. Let me give you an example of a damaged stretch of DNA. So this right here says a thymine dimer distorts the DNA molecule. Notice here this little bubble. But notice here, so when it says the dimer here in that little bubble, I wish I could write it here, but right here, these are supposed to be T's, all right? So across here, these would be A's. Notice that the T's have a line between them. They're bonded together this way. So if I were to enlarge that picture, if I were to redraw it down over here, that bubble, all right, like this, all right? These are T's, all right, sticking out, and so the rest of it's like here, so that's, I'm just redrawing that picture there. This right here, this line right here that's binding between them is not supposed to be there. They're not supposed to bind this way. They're just supposed to hydrogen bond with the bases across from them. And that's causing a distortion in the shape of the DNA molecules. So that's not good. So therefore, we want to, what our cells will do is notice that's not good. And then there's an enzyme called nuclease that will cut it out. So like right here, it cuts right here to right here. All right? It actually cuts out the damaged piece. And then we already have naturally DNA polymerase III that will add nucleotides. So it'll add the right nucleotides, so the right T's that aren't bonded the other way. And then DNA ligase will glue them all together, and we've got naturally fixed DNA. So therefore, like when I think I talked about when we talked about meiosis or mitosis, and you go out in the sun and your skin gets darker, that's already an indication that the UV light of the sun has damaged your DNA. Usually that's not usually that doesn't cause a problem because we have enzymes that can fix it, and this is how they would do that, all right? And so therefore, they would fix damaged DNA and repair them, and so, so that is one way to do that. All right, so that's if something's wrong with the structure or mismatch repair. One other last thing is that I want to talk about here is the replicating the ends of the DNA molecules. And so, so the ends are special. All right, let's talk about why we need to talk about them. So limitations of DNA polymerase create problems for linear DNA of eukaryotic chromosomes. So that's like our chromosomes. Our chromosomes are linear, our DNA is linear. The usual replication machinery provides no way to complete the 5' ends, so repeated rounds of replication produce shorter DNA molecules. Let me show you what I mean by this. I'm going to draw a picture here. So here's your double-stranded piece of DNA. And let's say this is the 5' end, this is the 3', so this is the 3', this is the 5'. So they're anti-parallel, okay? So when DNA replication occurs, all right, so helicase is going to unwind it, unzip it, and so on. So I'm just going to draw it here like this, all right? So here's my 5', 3', 3', 5'. All right, so the new DNA strand that I'm going to build, can you see that this is going to be the 3' end of this one, and this is going to be the 5'? Follow me? And then 5' and 3' to build my two new strands. So do you see that here, the new strand has to be built in the 5' to 3' direction. So this one's going to be built down this way. This one's going to be built up this way. Following me? All right, you need a primer. So I'm going to add a primer, all right, to start that off. So that's an RNA primer. Then, now DNA polymerase can add nucleotides, and we can go to the end here, add nucleotides, and go to the end here, all right? But remember then, what happens next is we don't keep the RNA primer, all right? So we get rid of the primer. So notice here, so I get rid of the primer, DNA polymerase can't add nucleotides here because it has to have a pre-existing nucleotide. All right, that was the whole purpose of the primer to begin with, is because we can't add it to the ends. So when we get rid of the RNA, guess what? Our ends just got shorter. All right, and then what happens is now, let's say that's the first time this cell replicates, because remember why we replicate is for cell division. Now, these two cells have shorter pieces of DNA. Now, what are these going to do? They're going to divide again. So now watch this, what happens if I take just this one right here, all right, and divide it. Do you see that now when these separate, this one, go down here, this one here is going to be a little shorter, all right? So this one's going to be a little shorter. It's missing that part right there. This one's okay, all right? This one will still be fine. All right, whoops, I got to make it farther apart here. This one will still be fine. It says 3', 5', all right? And so then this would be, for this one, the 5' and the 3'. All right, so it's going to be shorter. And so then when replication occurs, it's going to add the primer here, because this is going to be the 5' end, 3'. But then when it replicates, it's only going to replicate to the end here. So I'm going to have this piece of DNA is shorter. Do you see that? And then when this, then when I get rid of the primer here, that end is shorter. When this goes to replicate again, it's going to get shorter and shorter and shorter. So every single time your cells divide, the ends of your DNA get shorter and shorter and shorter and shorter. Do you see where that could be a problem eventually? Eventually, it could go into your genes, which are making your proteins, and that can be a problem. So in our cells, the ends, we have a little bit of a buffer with that, because the ends, this picture just shows you what I was drawing here. I want to show you here. The ends have a sequence called a telomere. So eukaryotic chromosomal DNA molecules have at their ends nucleotide sequences called telomeres. What this means is that they don't code for, they're not genes. So therefore, let's say if this is your piece of DNA, let's say your first gene, these ends don't code for anything. They don't do anything for us. So here's from right here is gene 1, and here's gene 2, and so on. And the ends don't code for anything. So do you see that when replication occurs and the ends get shorter, if they don't code for making proteins, they don't hurt you then, all right? So therefore, these ends that are just kind of gibberish, the A's, T's, C's, and G's that don't code for making proteins, they don't do anything for us. It's okay for a while if they get shorter, all right, as our cells divide. And this can become a problem the more the cells divide, because eventually you can start to, as the chromosomes get shorter and shorter with every cell division, it can eat into those genes. So this is what they think what happens as people age, all right? The older people are, the more your cells have divided, right? And the more your cells have divided, the shorter and shorter and shorter and shorter your DNA gets. And so therefore it can become a problem, all right? So they're doing a lot of research on these telomeres and how do we stop them from getting shorter, all right? And therefore can stop the aging process. So these telomeres don't prevent the shortening, but they postpone the erosion of genes because the telomeres themselves aren't part of a gene. All right, so it's okay if we don't have them. All right, but eventually, the more our cells divide, the shorter and shorter those ends get. And so this is a real picture of some chromosomes with fluorescent tags on them, and these are the telomeres at the ends of them. All right, and so the problem becomes, if you can imagine, is when we make egg and sperm cells. So the older you get, if your telomeres are shorter, we don't want to pass on shortened chromosomes to our kids. So if chromosomes of germ cells are the cells at the beginning of meiosis that produce egg and sperm cells, so they're in the ovaries of females and in the testes of males. If chromosomes of germ cells became shorter in every cell cycle, eventually essential genes would be missing from the gametes and you'd be passing on damaged chromosomes to your kids. That's not good. All right, so germ cells are the only cells that have an active enzyme in our body called telomerase. And telomerase, what that does is lengthens the telomeres. So even though as a parent you're older and your telomeres are shorter, it elongates them in the germ cells so that when you make your gametes, your kids are going to have full-length chromosomes. All right, and so therefore there's a lot of study being done with telomerase that how do we make it active in other cells and elongate our telomeres and therefore kind of prevent or stop or delay the aging process, all right, of our cells. All right?
I really like
All right.